Development of neuronal circuitry in the human prefrontal cortex during the midfetal and late fetal period. Structural organization. Transient fetal lamination of the prefrontal cortex, figure 2.1d, is characterized by prominent SP subplate, thick and consolidated CP cortical plate, and the presence of transient sublayers in the MZ marginal zone. The SP subplate contains an abundant extracellular matrix, quote-unquote weighting the lamocortical fibers, and relatively mature polymorphic neurons of quite variable morphology. Transient midfetal zones are easily visualized by both in vitro and in vivo magnetic resonance imaging. The most prominent transient zone is the subplate SP, easily delineated on MRI scans owing to the presence of abundant and hydrophilic extracellular matrix. During the midfetal and late fetal period of the prefrontal cortical areas, as cytoarchitectonically defined in the adult brain by Brodmann, 1909, or von Economo and Koskinas, 1925, still do not display two defining features. That is, granular layer 4 and large pyramidal neurons in sublayer 3C. At the beginning of this period, layer 3 neurons are not even born. In addition, the prominent SP subplate determines the lamination pattern, which is very different from that of the postnatal cortex. However, architectonic differences between major prefrontal cortical regions, dorsolateral, orbitomedial, and orbitolateral, are already visible on nissl-stained histological sections. Transient fetal zones display regional differences on in vitro magnetic resonance images of formalin-fixed brains, as well as on in vivo in utero magnetic resonance images. The dorsolateral prefrontal region shows two characteristic features: very thick subplate SP and tangential waves of migratory neurons in the superficial part of the SP subplate just below the CP cortical plate. Neurogenic events. During the midfetal period, neurons and glial cells are continuously produced in ventricular and subventricular zones. The subventricular zone is highly developed in the primate brain and seems to be a source of interneurons and glial cells. The overall increase in the number of mitotic cycles seems to represent the cellular basis of prefrontal cortex expansion during human evolution and development. As neurons are continuously produced, they also continue to migrate through the intermediate zone and SP subplate. Neurons migrating during the mid-fetal period are destined to supergranular layers and probably correspond to prospective pyramidal neurons of layer 3 that give rise to associative and commissural cortical pathways. The major pattern of migration of principal pyramidal neurons is radial migration along radial glial cells. This mechanism is particularly important for the thick cerebral wall of the midfetal primate and human brain, where neurons have to travel for a long distance from the site of origin in the ventricular subventricular zones to their final destination in superficial cortical layers. For example, in the midfetal human prefrontal cortex, radially migrating neurons have to traverse 5 to 9 millimeter thick SP subplate zone. As seen on nissl-stained histological sections, massive waves of migratory neurons together with rows of glial cells form a transient sublayer within the superficial SP subplate, figure 2.1d, of the human prefrontal cortex, which was first noted by von Economo and Koskinas, 1925. During the midfetal period, the growth of subcortical, subcortico-cortical, and cortico-subcortical projection pathways reaches its peak intensity. The lamocortical pathways grow through the intermediate zone during the pathway selection stage, figure 2.2a, and through this subplate SP during the regional target selection stage. The major input for the prefrontal cortex may be traced by selective histochemical staining of growing fiber tracts. For example, figure 2.3a, 2.3b. The growth of thalamocortical pathways partially overlaps with continuous growth of afferent fibers from basal forebrain in the external capsule. Fibers from the amygdala were not directly visualized, but this nuclear complex matures very early, and it is very likely that projection to orbitomedial cortex matures in parallel with thalamic projection. Cortico-subcortical pathways from the prefrontal cortex are more difficult to trace in the human brain. In the rhesus monkey, developing corticostriatal projections are in spatial register with the striatal, cytoarchitectonic, and acetylcholinesterase (ACHE) rich units. Goldman-Rakesh, 1981. In the human striatum, these units develop during mid-gestation, indicating that mid-gestation represents a developmental window for growth and establishment of human prefrontal cortical striatal pathways. The growth of other subcortical efferent pathways, for example, long cortical pontine and corticospinal axons of layer 5 pyramidal neurons, is less well known. Two lines of evidence suggest that subcortical efferent pathways may have reached their target structures or levels. First, microtubule maturation in cell bodies of prefrontal layer 5 is accelerated after 18 postconceptional weeks, and expression of specific genes seems to be required for proper targeting of their axons. Second, corticospinal fibers originating in the human frontal motor region arrive in the spinal cord already at 24 postconceptional weeks. Neuronal circuitry and function. The most differentiated neurons of the mid-fetal frontal cortex are polymorphic SP subplate neurons and Cajal-Retzius neurons of the MZ marginal zone. The number of synapses has increased in the subplate and marginal zone SP and MZ, and transient circuitry of the SP subplate displays remarkable complexity. There is electron microscopy evidence that SP subplate neurons are postsynaptic elements since synapses are readily found on their proximal dendrites and cell bodies. The vast majority of synapses are asymmetric and therefore probably excitatory. Subplate neurons display a striking variety of neuronal morphologies. As shown in rhesus monkeys, Meinecke and Rakesh, 1992, and carnivores in Tonini and Schatz, 1990, many SP subplate neurons synthesize GABA, while approximately 60% of subplate SP neurons may synthesize glutamate. Presynaptic elements for SP subplate circuitry originate from monoaminergic and cholinergic afferents in other SP subplate neurons, while thalamic axons and axons of neurons residing in the CP cortical plate may represent an additional presynaptic input. As a rule, GABAergic neurons co-express various neuropeptides. To summarize, rich presynaptic input from modulatory nonsensory sources impinges upon a complex transient population of neurons. Intrinsic neurons are interconnected. Thus, one network communicates with external input, while the other network is endogenous and very likely oscillatory.